So, good morning, grade 12 students. In this session, we're going to continue with our chapter about the nervous system, the neurophysiology, in chapter 12, the neurotransmitters and the medical applications, page 134 on your book. Yesterday, we started about the document one, the neurotransmitters and the membrane channels. We talk about the neurotransmitter. We have two different groups. We have the classical neurotransmitter and the neuropeptide. The classical neurotransmitter, they include either amino acid or a derivative of amino acid. So we started with this different types or different groups. The classical neurotransmitter, they include an amino acid or a derivative, whereas the neuropeptides, they are found of many amino acids, where according to their function, the neurotransmitter, they are classified either excitatory or inhibitory neurotransmitter. We said that GABA and glycine, they are exclusively, they are inhibitory neurotransmitters. Why? Because their receptors always correspond to the Cl- channels, to the chloride ions channels. These will cause the hyperpolarization in Hasselhoff. Whereas the acetylcholine is generally excitatory, where in the case of the skeletal muscles, whereas it's inhibitory in the case of the cardiac muscle. I also we talk about the inotropic and the metabotropic. Inotropic, they have an instant effect and they have a rapid response, whereas the metabotropic, they have the second messengers and they take longer time to response, and this response will be a permanent effect. We talk about the acetylcholine, they are involved in the movement. We talk about the serotonin that's involved in the temperature regulation. And also we talk about the GABA, it has inhibitory role and involved in the inhibitory synapses. We talk about the substance P, the pain substance. It is responsible for the transmission of the pain messages, as well as we talk about the endorphins and the enkephalins. All of them, they are analgesic and they have a role in decreasing the feeling of pain. Also, we have a drug that has a role in the decreasing the feeling of pain or inhibition of the feeling of pain by inhibition of the exocytosis of the P substance. This is called the morphine. So here we talk about all these neurotransmitters and we saw probing the documents in the previous session. Now we're going to move to document 4. We're going to talk about the action of drugs on the synapses. So for a start, we have two different phases of the nervous system. We have either the amplification of the nervous system, we have either the excitation of the nervous system, or the inhibition of the nervous system. Talk about the amplification, we have first to amplify the nervous system, to excite the nervous system, to have excitation, we have to provoke the exocytosis of the neurotransmitter from the secretory vesicles in the presynaptic terminal. So whenever the neurotransmitter they undergo exocytosis, they are released from the secretory vesicles in the presynaptic terminal. So this will lead to the amplification of the nervous system. Second, we have to inhibit the recapture of the neurotransmitter. As we said before, neurotransmitter, they are going to be either recaptured or they are going to be degraded by specific enzymes. So if we inhibit the recapture of these neurotransmitters in the presynaptic terminal, so the action of this neurotransmitter will continue and there is no stop for this action, and this will lead also to the amplification of the nervous system. In addition to this, what do we have? We have to inhibit, if we have inhibition of the action of the enzyme that degrade the neurotransmitter. As I said before, neurotransmitter is going to be released from the presynaptic terminal into the synaptic cleft. Whenever it binds to its receptor, it leads to the facilitating of the action, it leads to the excitation or whatever. This will lead finally to what? This will lead to finally to have the reaction or to have the action potential or to have the membrane potential and to lead to the response. Then these neurotransmitters, they should be recaptured or degraded. So whenever they are recaptured or degraded, so the action of this neurotransmitter will stop and is going to be inhibited. But if we inhibit the recapture of these neurotransmitters or we inhibit the degradation of these neurotransmitters by specific enzymes, so this will lead to the amplification of the nervous system. Number 4, to facilitate the binding of the neurotransmitter to its receptor in the postsynaptic terminal. So whenever the neurotransmitter binds to its receptor on the postsynaptic terminal, this will lead to its amplification. Amplification of what? Of the nervous system. So we have four important steps or four important phenomena that occur that leads to the amplification of the nervous system. The first one, I will repeat them. The first one is to provoke the exocytosis to help to facilitate, to let's say, to make it easier, the process of the exocytosis of the neurotransmitter from the presynaptic terminal. This is the first one. Second one, to inhibit the recapture, of the enzymes, or sorry, of the neurotransmitter and the presynaptic terminal. This is the second one. So first one, to provoke the exocytosis, the release into the synaptic cleft from the presynaptic terminal. Second one, to inhibit the recapture, also to inhibit their degradation by inhibiting the action of the enzyme. And also we will have to facilitate the binding of the neurotransmitter to its receptor and the postsynaptic. All of these, they lead to the amplification of the nervous system. However, inhibition of the nervous system, they are opposite to those in the case of the amplification. And the amplification, we have to provoke the exocytosis of the neurotransmitter from the presynaptic terminal. In the inhibition, we have to inhibit the exocytosis of the neurotransmitter from the presynaptic terminal. So we have to inhibit the exocytosis of the neurotransmitter from the presynaptic terminal. Second one, to provoke the recapture of the neurotransmitter and the presynaptic terminal. So you have to help the recapture of the enzyme of the neurotransmitter in the presynaptic terminal. This is the second one. While in the case of amplification, you have to do what? You have to prevent or to inhibit the recapture. Third, in order to lead to the inhibition of the nervous system, you have to provoke or to facilitate or to help or to allow the action of the enzyme that lead to the degradation of the neurotransmitter. So as you see, they are opposite to those in the case of amplification. And the fourth one, to inhibit their binding to the receptors. So here, if you inhibit the binding of the neurotransmitters to the receptors, this will lead to the inhibition of the nervous message. Whereas in the case of amplification, you have to allow or facilitate or provoke the binding of the neurotransmitter to their receptors. So as I said, these hints, they are very important, and these will help you a lot whenever you are interpretation or you have conclusion or you have, for example, a hypothesis. So these will help you. In the case of the amplification of the nervous system, we have amplification. So you have four important tips or four important phenomena that occurs. Opposite of them, they occur in the case of the inhibition of the nervous message. For example, if the neurotransmitter action, for example, elongates, for example, or if it stays for a longer time. So this maybe can be described by what? By inhibition of the degradation by this neurotransmitter by the enzyme, or the inhibition of the recapture, or you can say it's this function, it's allowed it to bind to the receptors for a longer period of time. So this will help you to conclusions, hypothesis, and also for the interpretation. So these notes are very important in the case of inhibition of the nervous system or in the case of the amplification of the nervous system. Now let's move to document 4, action of drugs on the synapses. A drug is a psychoactive product that affects the behavior of the person by causing a sensation of evasion of euphoria. It can, within certain consumption values, lead to serious problems and might cause death. How does this happen? Before we start, let's remember together what the meaning of agonistic and antagonistic substances. Agonistic and antagonistic. What do you think, Gabadia? What the meaning of agonistic and antagonistic substances? If I say
Uh,
antagonistic substances, what does this mean?
Antagonistic is the opposite of what the body does.
So they have opposite function. So these
Function.
drugs or these substances, they have opposite function. Wherever agonistic, they have the same function, or at least they facilitate the function of each other. So this is the meaning of agonistic. While antagonistic, they have opposite function, or at least it inhibits its function. Okay, so this is antagonistic and agonistic substances. Already we covered them before. So various drugs, they act at different levels in the synapse. They can provoke the functional modification. Curer, we talk about the curer. Curer is an alkaloid of plant origin. It's a paralyzing substance that was used by Amazon Indians who used to apply it to the tips of the rose. To the tips of the rose, they use the rose in order to capture their prey. So they put on the tips of the rose, they put this substance curer. This curer, they can be used to paralyze the animals so that they can catch their prey easily whenever it's affected by this substance, which is the curer. Nowadays, curer it's used in the surgeries. We can use it in the surgeries by to cause the relaxation of the muscle in the patients. So as I said, curer, alkaloid of plant origin. It's a paralyzing substance that was used by the Amazon Indians who used to apply it to the tip of the rose to capture their prey. Purer products are nowadays used by surgeons to cause muscular relaxation in the patients. In the last century, the physiologist Claude Bernard. Claude Bernard is one of the most famous physiologists that work at the level of the neurophysiology. So he did a series of experiments to show the action of this drug in a neuromuscular synapse. Neuromuscular synapse has receptor for acetylcholine. You have to know that curare and acetylcholine they have highly similar structure that we said before, highly similar spatial configuration. So they are very similar structure. Their molecule they have very similar structure. So their structure is very similar to each other, and this allow them to occupy the same sites on the receptor. Since they have the same structure, the same, for example, molecular structure or the same spatial configuration, so they can bind to the same receptors. However, cocaine, which is an alkaloid extracted from cola leaves, interferes with dopamine as well as noradrenaline. These neurotransmitters, they are usually reabsorbed by the neurons that release them. What does cocaine do? Cocaine, it blocks the reabsorption leading to excess dopamine. Remember when we talk about the cocaine, it inhibits the recapture. So review this. So it inhibits the recapture for the dopamine. So it blocks the reabsorption, the recapture, leading to excess dopamine and noradrenaline in the dopaminergic and adrenergic synaptic graph. So cocaine, it has an important role. It inhibits or it prevents the recapture of the dopamine. It inhibits the recapture of dopamine and noradrenaline. This also cocaine it acts on the cell membrane and blocks the propagation of the nerve impulse by opposing the Na plus ions channel. So here, moving to experiment and document A. Document A talking about the results of the experiment of Claude Bernard. So results of the experiment of Claude Bernard, what happens here? Here we have injection of curare into the blood. We said that curare, it has a role in the relaxation of the muscle and even it leads to the paralysis. Here we have, this is the sciatic nerve, okay? And here we have the ligature that blocks the circulation and leaves the sciatic nerve free. First of all, we have right paw and the left paw. First, in the first experiment, what did they do? The first experiment, they did stimulation of the sciatic nerve of the left paw. So stimulation of the sciatic nerve of the left pole. What happens? We have extension of the pole extended. So stimulation of that sciatic nerve of the left pole. So what happens? The extension of the pole occurs. Whereas the stimulation of the sciatic nerve of the right pole, it leads to no response. So this leads to no response. However, the direct stimulation of the gastrocnemius muscle, gastrocnemius occurs here at this region, so the gastrocnemius muscle, this will lead to the extension of the pole. So here, stimulation of the gastrocnemius muscle leads to the extension of the pole. How can we explain this? Step by step. So I'm going to interpret. This is part one. They said, why don't the experiment of the Claude Bernard verify the mode of action of curare and acetylcholine receptors and so on. So I'm going to replace this by interpret the experiments of document A. So what did they do? First, they did stimulation of the sciatic nerve of the ligated left pole. Ligated, we have a ligature. So we have ligature رابطينها مثل كأنه رابطين أو شادين عليها. So we have ligated left pole. What happens? So ligated left pole, we have the pole extends. While after stimulation, the sciatic nerve of the right pole, there is no response, there is no extension of the pole. So what does this show? This shows that the curare is transported via blood, and curare, it stops the extension for the pole in response to the stimulation. Either it works at the level of the nerve, or it works at the level of the muscle, or it works at the level of the motor and the plate, which is the neuromuscular synapse. Here, as we said before. While after we did the stimulation of the gastrocnemius muscle of the right pole, what happens? The pole, it extends. So we have extension of the right pole. So what does this show? This shows that, or this indicates that curare doesn't act directly on the muscle. So it can act through other muscles. So in addition to the interpretation, here this experiment doesn't prove if curare affect the sciatic nerve or it affect the synapse itself, which is the neuromuscular synapse. Here in this question, we don't have if the curare it affects the nerve itself, which is the sciatic nerve, or it affects the level of the synapse, which is the motor and the plate or the neuromuscular synapse. The first experiment, what do we have to do? They said you have to propose an experiment. So proposing an experiment to show if the curare mode of action it's on the acetylcholine receptors or not. So to do this, we have to do about 3 experiments. First, the motor and the plate of the muscle is isolated, meaning that the neuromuscular synapse is going to be isolated. Remember when we talk about the motor and the plate, neuromuscular synapse and the neuroglandular, we call this neuromuscular and neuromuscular, we call it the motor and the plate. So here we have to isolate it, we have to separate it, and then we have to inject it with acetylcholine into the synaptic cleft. The muscle, what will happen? The muscle will contract due to the action of the acetylcholine. And the second experiment we have to do, you have to inject curare into the synaptic cleft. And then you have injection by the curare. The muscle does not respond. So this means that curare blocks the action of acetylcholine. So here you have to put, instead of curare, you have to put ACH, followed by injection of, you have to say acetylcholine is injected, and in the synaptic following by injection of curare. This indicates that the curare blocks the action of the acetylcholine at the level of the synapse. While the third experiment, we have after the injection of radioactive curare, radioactivity is observed lining the post-synaptic membrane. So this can be explained that the curare can bind to the receptors of acetylcholine on the post-synaptic membrane. So they have highly similar spatial configuration, and this will lead to the binding to the receptors on the post-synaptic terminal. So here, you have to put instead of curer, curer is injected in the synaptic trap, followed by injection of curer. So here you have to put acetylcholine instead of this curer. So here, curer is injected, you have to put acetylcholine is injected, and then followed by injection of the curer. So here, this experiment will help you to identify the mode of action of the curer and the acetylcholine receptors. As we said, they have highly similar respiration configuration, and the curer will bind to the receptors of the acetylcholine and prevent their working. That's why they can work and can bind to this. Here we have the action of amphetamine. Here they have a highly stimulating anti-fatigue effect. Intoxication by amphetamine is possible in case of injection. Euphoria is expressed in a brutal, intensive manner, but it's quickly by intolerable one. What will happen? Amphetamine, they are excellent drugs that stops the appetite, release dopamine from the storage vesicles, even in the absence of any presynaptic message. So here, the amphetamine, they have an important role and they work at the level of the synapse. Also, we have benzodiazepines and anxiolytic drugs, anti-anxiety drugs such as the Valium and Francine اللي بيكون عندهم أعصاب ياخدوا Valium ويبقى عندهم كآبة and this activate the fixation of the GABA on the receptors of this synapses. So here, the GABA has an important role in the inhibition of the nervous message. So what I want from you till now, I want you to hear to understand the function, the mode of action of the agonistic and the antagonistic drug. You have to know that both acetylcholine and the curer, they are antagonistic. They have opposite function and they have different function. One of them leads to the excitation and the contraction of the muscle, which is the acetylcholine. However, the curer leads to the relaxation of the muscle and as well as leads to the relaxation of the muscle and even it leads to the paralysis. You have to know that cocaine, it has a role in blockage of the recapture of the neurotransmitters and it blocks the recapture of dopamine. That's why we have a prolonged effect of the dopamine in the case of cocaine injection. And these are the important notes for this session. Next session, we're going to continue to talk about amphetamine in details, and we're going to talk about the difference between addiction and dependence, or the difference between addiction on drug or dependence on drugs, tolerance and addiction and dependence. Also, we're going to talk about the medical applications and to talk about the different mode of action of the drugs, how do you use them and why did you use them. So in next session, we're going to continue with this document. So your job for this is to copy only, interpret the experiment of document A, and this is the proposed experiment, first, second, and third experiment. Okay, this is action. And part B, justify the following statement. Curer and acetylcholine, they are antagonistic substances. Why they are antagonistic? Morphine and enkephalin, they are agonistic. Why they are agonistic? So here, first of all, you have to know that acetylcholine is excitatory at the level of the neuromuscular synapse. It has an excitatory role. The opposite of this, to the contrary, curer has an inhibitory role. It blocks the response for the muscles. So it's inhibitory. Therefore, the acetylcholine and the curer, they have highly similar structure and they bind the same receptors, but they have different functions. They have similar structure, but they have different functions. That's why they are antagonistic. In the parent circuit, if you remember, when we talk about morphine, morphine binds to the same receptor of the enkephalin, but morphine has a role in the total inhibition of the exocytosis of the P substance. So morphine, it inhibits totally the exocytosis of the P substance. However, the enkephalin, it's an endogenous substance. It leads to the partial inhibition of the P substance. So both of them, they have the same function, even though not the same effect. But both of them, they have the same function. That's why we said they are agonistic. So as a conclusion, curer, it causes the relaxation of the muscle. However, Acetylcholine causes the contraction of the muscle. So they are antagonistic. While enkephalin and morphine, they inhibit the pain circuits by inhibiting the exocytosis of the pain substance, the P substance, but sure with different levels. So enkephalin, it inhibits partially the exocytosis of the P substance. However, the morphine, it inhibits totally. So both of them, they have the same function. We can say about them they are agonists. So till now, we solved part one. This is part one. Instead of why, I said I made it an interpret in this part. And in the second part B, design experiment, we designed an experiment to prove the mode of action. Part two, we saw part A, curare and acetylcholine, they are antagonistic. So we said why they are antagonistic. And also morphine and enkephalin, we said why they are agonistic. Tomorrow, on the next session, we're going to continue with this lesson with document 4, talking about the drugs and their mode of action of drugs on synapses.